It's been time to move along in the program, and the next company to present is Immunicum, and here to present the company is Simon Seilermaker, who is COO of Immunicum. Take it away, Simon.
Thank you, Klaas. I'm here to tell a bit about Immunicum. Immunicum is a cell-based therapy company who is listed on Nasdaq Stockholm, so I'm obliged to show you this disclaimer. Our cell-based therapy is in the phase 2 development, so that means that we've actually completed now a phase 2 controlled study in kidney cancer, and that study has a next survival update planned for mid of this year. Apart from this study, we've completed actually a number of clinical studies and treated already over 90 patients in different indications, and have a very good safety profile, which is also very important in this space of advanced therapies. Our cell therapy is currently being produced in Germany at a GMP level facility, but we're also initiating commercial scale activities with Hitachi, which is a US-based manufacturing organization. Recently, and as recent as last month actually, we received an RMAT designation from the FDA. That is a Regenerative Medicine Advanced Therapy Designation. A very new program that the FDA has announced, but very similar to one they already had, which is the Breakthrough Therapy Designation. And that's a program that is helping selected therapies that the FDA thinks are of interest, helping them to move through development much more smoother by giving them much more guidance. So in our case, we've received the designation last month based on our phase 2 data. And we will now have a more ongoing dialogue with the FDA to see how we can best design our next development steps. Also, actually in 2018, we received an ATMP certification from the European authorities that is also looking at part of our regulatory file. So there's a lot of regulatory validation already with the different authorities with our cell therapy. Apart from regulatory, in 2018, we also announced with industry a collaboration with Merck and Pfizer. This is for the use of their checkpoint inhibitor in combination with our therapy in the phase 2 part of an ongoing study. So we're currently in phase 1 for the Iliad Study, and in the phase 2, we will start using their checkpoint inhibitor. But this is also something they only give to selected therapies, so also shows their interest in our program. Now, from a financial perspective, as I said, we are listed at the Nasdaq Stockholm, the main market. In December 2018, we were able to complete a relatively big financing round of about 350 million SEK. And this really helps us to be financed until the end of 2021 through our ongoing development programs that are currently, for example, with Iliad ongoing in phase 1b and the Bolagsdag study in survival update for phase 2. And with that, we have also very much a stable situation in the current ongoing pandemic. From a team perspective, we have our CSO, who's the founder of the company, who is the acting CEO at the moment while we are looking for a permanent CEO position. Around him, we have very complementary expertise in terms of the regulatory side of the business, the production side of the business, and I'm helping, for example, a lot on business development, corporate development, and investor relations. Now, going into the product that we are developing, it's a unique off-the-shelf immune primer. Now, starting with the off-the-shelf, that means that we actually use cells from a healthy donor. So any healthy donor, similar to a blood donor, can donate immune cells. And these immune cells, we take and activate them to turn them into inflammatory dendritic cells. Now, dendritic cells have mostly been used as a vaccine in the past. We are not doing that. We are using them as an allogeneic immune primer. And that means that we inject these inflammatory cells into the tumor tissue of a patient, so inside their body. And that creates this immune response against that tumor tissue inside the patient. And because it's inside the patient, it is a tumor-specific response. And that's very crucial if you want to do something with immunotherapies. It has to be tumor-specific. And we are achieving that by injecting our cells into the tumor of a patient. And thereby, we are an optimal combination therapy to other therapies that can further help the immune system from attacking the cancer. And checkpoint inhibitors are one example, but also other targeted therapies or even standard cancer therapies like chemotherapies. Now, from a production perspective, from one healthy donor, we get about 100 doses of our cells. So it's not one donor, one patient. We can actually treat about 50 patients from one donor. And these cells we can store for 4 years in the freezer. And that also allows us to produce our cells and at any point in time use them to treat a patient. And that's really what makes it off the shelf. We can use it at any time for any patient that is that you can inject into the tumor. Now, in terms of the landscape, we are an immune primer, which really means that we're activating the immune response, and then we act in synergy with agents that are then blocking the tumor's defense against the immune system. So checkpoint inhibitors are a very good example there. They're very effective if there's already an activated immune system that is not able to kill the cancer, and they block the tumor from preventing that. However, in the majority of patients, you don't have a sufficiently activated immune system. So we are really there to activate and boost the immune system, and therefore we work in synergy with agents that can really help attack the cancer cells. So it's really a combination therapy. We are not intended to be used on our own, really as a combination therapy to get the most out of the immune response. More recently, we have been looking at the preclinical efficacy of our therapy in combination with different classes of other therapies that I just described. PD-1 is a class of checkpoint inhibitors that are used a lot now in different cancer indications. And we've previously been shown that on top of PD-1, we can nicely delay and have an impact on the tumor growth of in this animal model. But also a more, I would say, interesting class is the CTLA-4 class that is used in some tumor indications. On its own, you can see here on the left, it is already quite active, and you can see the delay in the tumor growth that it already has as a monotherapy. If we add 2 injections of ilixadencel on top of CTLA-4, we really saw that we induced 7 out of 10 animals into a complete response. So the tumor completely disappeared. And even when we later re-challenged these mice, so we gave them another injection of the tumor was actually unable to grow. So there was still a complete response happening, even when we gave them another challenge of the tumor without giving further treatment. And this really shows that you've induced a systemic immune response that is able to target the cancer. Now, this is a very interesting combination. And as you can see in our pipeline, we are trying to position ilixadencel in different types of combinations because every solid tumor currently has different standard of care. And also every tumor has a different type of sensitivity to immunotherapies. So for example, in kidney cancer, we have completed a phase 2 with kinase inhibitors, which is a standard anti-cancer agent. But in our new study, the Iliad study, we are combining with checkpoint inhibitors in indications where only checkpoint inhibitors are approved, head and neck, non-small cell lung cancer, and gastric cancer. But even in those situations, the checkpoint inhibitor alone is only effective for a small proportion of patients, and we try to improve that. And also for future indications and studies, we will be looking at the right combination in every setting. And the big benefit that we have as a therapy is that we can be combined with different classes, and that we have a very good safety profile, so that we also don't cause issues to the patient with combining with different therapies that tend to have more toxicity issues. Now, going into the clinical studies that we have completed, 4 to date. It all started with a small phase 1 study in kidney cancer here in Sweden. 6 patients treated as a monotherapy, and then 6 patients who received the standard of care later on. And there actually we saw very positive signs of the mechanism of action, but also survival benefit and responses happening, especially in these combinations. And that really helped us drive this hypothesis that the combination with ilixadencel on top of a standard therapy can really help get the most out of the immune response. So based on that study, which was published in 2017 in the end, we actually started in 2015 a big phase 2 study in 88 patients that we have now completed last year and are currently in the survival follow-up. In the meantime, we've also completed a phase 1 in liver cancer and a phase 1 in GIST, which is a smaller indication, rare indication. And also those studies really gave us a lot of comfort in terms of what are the effects that we are seeing with ilixadencel, what are the right combinations to make in these different indications. And also there we are looking for ways to move it forward in the right combination. Now, to tell you a bit more about the phase 2 study, as I said, this was a study in 88 patients, and it was a randomized, controlled, but exploratory phase 2 study. Well, this means that we had 2 groups, one control arm that received the standard therapy, and then the active arm with ilixadencel on top of the standard cancer therapy. The exploratory part of it was that we had different endpoints included, and at the end of the study you look at, okay, what are the effects that we are observing and what can we use for our next study to get to the market. Now, these were patients with newly diagnosed synchronous metastatic RCC, which means that they are newly diagnosed with kidney cancer but already have metastases at the diagnosis. This means that they would normally have their kidney removed that contains the kidney tumor, and then start the treatment for their metastatic disease with sunitinib. So in our study, we had these patients enrolled. They would be randomized to either receive 2 shots of ilixadencel before the surgery into the kidney tumor or no injections. Then both groups would have their kidney removed, and then a number of weeks later, you could start with the sunitinib treatment. Now, because of this waiting period for the surgery and this waiting period for starting the standard of care, in this fast progressing disease, you already lose quite a number of patients. So in both groups, we lost about 18% of patients due to fast progression, not even being able to start with the standard of care. This led us to have, for survival, we followed all patients, and all patients are included in our survival overview. But for tumor response, where you're measuring the tumor size and seeing if they decrease, we actually had 45 and 25 evaluable patients that actually received the standard of care. Now, let me show you what is currently in the survival follow-up. So when we first read out this study in August last year, we only looked at patients that have been followed for at least 18 months. And after 18 months, we didn't see a difference yet, likely because of patients progressing relatively quick in the beginning. And still a lot of patients that are not responding to either therapy, the standard of care or ilixadencel. However, what we see happening is that patients that have been followed for a longer duration, especially patients that had a good response, tend to have this response for a much longer duration than the control group. And also the survival starts showing that. So we have patients in this study that have already been treated for 4.5 years, followed for 4.5 years, and are still alive, while in the control group you see more patients dying at a certain time point in follow-up. We can show that a bit on the next slide, where we show the 2 groups side to side. We have more long-term survivors in our group as compared to the control group. And this is really showing what immunotherapies have been showing in other indications, where if a patient is responding to an immunotherapy, they tend to have a much better survival as compared to the standard cancer therapy that they may respond to, but then still progress when there's, for example, resistance to the therapy. Now, part of the explanation behind this is that the group that is receiving sunitinib, especially on top of ilixadencel, had a very good response. So when we look at the confirmed response rate, which means that a tumor has to decrease in size and at the next scan still be decreased in size or more, you count them as a tumor response. And we had 42% in our group, and in the control group, only 24% of patients had an actual tumor that was decreasing over time. So this big difference really shows that, okay, the tumor is being attacked by the immune response in combination with the standard of care. And what we see if we compare that result to other agents in the kidney cancer space is that other immunotherapies tend to show a similar effect. So if you add an immunotherapy on top of the standard of care, you can increase the response rate and also increase the number of complete responses happening, which means that the tumor is completely disappeared. And that is very exciting in the field of immunotherapies, where you look at how many complete responses can you achieve. And that is something that is very exciting about our data. Not only have we increased the number of responses, but also the complete responses have been increased, where you really do not expect that on the standard of care with selitinib. So the fact that we're already in the same range as very active immunotherapy agents like nivolumab, ipilimumab, the PD-1 CTLA-4 combination is very promising for us. And that's why we're really now looking at the next step in development, how we can be added, for example, on top of these agents because of a good safety profile, or be combined with selitinib in a different setting because we've shown such a good combination effect. Now, quickly on the ongoing study with checkpoint inhibitors, we have the Iliad study ongoing. In the phase 1 part of that study, we combine with pembrolizumab or Keytruda, which is the standard checkpoint inhibitor that is approved in the US for these indications included. We've already completed the first dose cohort, which was a lower dose for our therapy. And now we are close to completing the second dose cohort, which is the normal dose level for ilixadencel. We should report on that at the end of Q2, so that is of course nearing. And then after that dose, we can open up the remainder of 15 patients and treat these 15 patients actually without any staggering in between. And that should give us results on of course, the safety, but also efficacy in the foreseeable future when we have treated those patients. Now, after that phase 1b is completed, then we actually have the collaboration with Merck and Pfizer to move into the phase 2 part of the study. And that's in combination with avelumab or Bavencio, which is their checkpoint inhibitor. Now, as a conclusion, and also my last slide for today, the immunotherapy effect that we are observing is very encouraging. We see complete responses happening and durable responses happening where you don't expect that to happen based on the standard cancer therapy. So we're really adding that on top of the therapy. And this is leading to the tail of the survival curve, this flattening of the curve where patients stay alive after having a response, while with the standard cancer therapy, at some point you progress and unfortunately die. This is very promising, but of course something that we have to continue following over time. So we will have actually the next survival update mid-2020, and every survival update that we will have on this study will further mature our thinking and the data about this long-term survival benefit. And that's very important also in our regulatory interactions. We've sent these results to the EMA and the FDA in the US. The FDA has given us this RMAT designation based on that data, so they already thought it was very promising. But we will continue to update them, and soon we will have a meeting actually with both authorities. We expect to receive feedback during this quarter. But that's receiving feedback, that's the first step. With the FDA, we can then have more ongoing dialogue to really design the next steps of development for kidney cancer. And as I said, for Iliad, we also close to getting a safety update from that study, and then we can open up to treat more patients in that study. So there's a lot happening in terms of current studies and future studies, and a lot of updates in the coming months on that. So I hope I've excited you for the company, and of course, if there's any questions, feel free to reach out to us.
Okay, thank you so much for the presentation, Simon.
Great.
And let me start off with a question about this recently achieved RMAT designation. Great work by the team, by the way. If you just could elaborate a little bit about how you think this will be influence what you need to show in a phase 3 trial with ilixadencel in renal cell carcinoma.
Yeah, so it's of course, it's going to be an ongoing dialogue on what their expectations are. We see especially the FDA becoming very innovative in terms of what they accept from new therapies, because they also know that with the pace of development of new therapies, they really have to keep up and cannot simply wait for the standard endpoints of survival. For example, in some of these indications, the survival is improving so much that the studies become longer and longer before you can really read out a certain difference. And that's where they for the RMAT designation specifically really emphasize that they would accept and recommend us to use surrogate endpoints for a next study to be acceptable for approval to the market. So in our case, we are expecting that our next study, you always want to look at survival, but that can be something that, for example, is reached already after you've been approved on the market, but you use a certain surrogate marker, for example, a durable response. And if you already see a difference there, they might approve our product based on an endpoint that is earlier in the study, so a shorter study duration, and we would already be approved to the market. And then later on, you would have this readout still of the survival. So we are expecting the next study to be shorter in duration, and therefore also of course the expectations in terms of what our organization has to do for such a study is a lot lower. And what we expect is that with the FDA, we can really discuss what endpoints are you looking for? What is the minimal safety database, for example? How many patients should we at least have treated to confirm that this is a safe drug that can be given to all patients in the future after market approval? And that really makes the chances of success of our next study a lot higher. Without this RMAT, you would have still one meeting in a way to confirm if they agree with your thoughts. You would start a study and you would hope that at the end of the study, they indeed agreed with the design and the assumptions of the study.
Great. And another question about the RMAT designation. This is from the web, and looking beyond the phase 3 trial and as a product. Do you believe this is a bit speculative, of course, but do you believe this will have impact on perhaps pricing and the willingness of reimbursements and such things?
Yeah, that's an excellent question, and indeed a bit speculative. Look, my belief is that the authorities in a way still look primarily at if you're truly better than what is currently the standard of care. And still in a reduced development timeline, like with the RMAT, you would still have to show that you're better, but you might use endpoints that are a bit softer than the survival endpoint. So the authorities mostly look at if you're truly better and if you're safe to give to patients. But it does, let's say, prioritize your product a bit. Having this designation as a stamp of approval does show that this is actually a therapy that the regulatory authorities believe can make a difference. And in the end, we have to show that we make a difference and show that, of course, to payers and insurances and governments that we should be reimbursed for this. But it does show that this is not a marginal benefit that we, for example, seen in our phase 2 already. And it has the potential to change the lives of patients. Complete responses is a very good example of that, of course. And in our discussions also with interviews with payers, we've also seen that, for example, complete responses is something they're very excited about, because a complete response also means that you can stop treatment and you don't have to give subsequent treatments that they also have to reimburse. So I think that's the exciting part. It's a bit of the stamp of approval that this can make a difference. And in the end, if our pivotal study, of course, shows that we have a substantial difference, then you will get reimbursed.
Great. And I guess RMAT is also important when it comes to partnering. And if you just could give us an update where you stand in this kind of process. Have you, what kind of activities are you doing right now?
Yeah, business development is an ongoing process. You continue updating the more important pharmaceutical companies on what you're doing. And you're always trying to find that sweet spot where they have need for your therapy and they have to act now. And you also need them at the right point to make the most out of the value of the program. And this is an ongoing dialogue. As you can imagine, when we had the first readout of the results, a lot of companies were interested, but they also wanted to hear what the authorities thought about this, because phase 2 is a crucial inflection point where you should have proof of concept of your therapy, but now it's the question, how does your last study look like? And that's what we have also been getting from partners, that they really would like to see the feedback from the authorities and in designing the next study, because likely if they are stepping in through, for example, collaboration or a license deal, they would be involved or even leading such a study. So that's very crucial for them. And as you can imagine, the RMAT designation has given them more excitement about the feedback from the authorities, because obviously at least the FDA was already quite enthusiastic about it.
Yeah, great. And my last question is a little bit more of a nerdy thing, and it's about your preclinical data that was presented in your presentation. I mean, it looks like a little bit like the combination with CTLA-4 gives better responses in tumor responses than if you combine it with PD-1s. What could be the explanation for this? Or is it just the modeling?
No, yeah, exactly. So, preclinical models are, of course, you're trying to have a similar situation as the human cancer situation, but it's never the same. In this specific model, I have to say that you wait for the tumor size to be at a certain point where, for example, PD-1 therapy actually is no longer effective. So if you just give PD-1, it's almost the same as giving a placebo at that point, because the tumor already has their defenses up, and there's no way for PD-1 therapy alone to be effective in that setting. CTLA-4 has a different approach, different pathway, and does have effects in that model. And then indeed, on top of CTLA-4, we see very promising signs. Now, when you look at the clinical situation, the human situation, we know that, for example, PD-1 is effective in a number of patients, but definitely not all. And CTLA-4, for example, it has been effective in melanoma on its own, but it's especially used as a combination to PD-1 in different indications that are very difficult to treat. So I think you can only take this as a mimic of perhaps a situation with indications that are not responding to checkpoint inhibitors, where CTLA-4 can make a difference, and elixir and cell seems to increase even that likelihood of getting an immunotherapy to work. So we don't see this as CTLA-4 being superior, but for very difficult to treat tumors, this might be a combination that we are looking at.
Thank you so much, Simon. Have a nice day.
Thank you. You too.